Okay, welcome back, everyone. Our next company to speak to us is BioChange. BioChange inventing a tissue scaffold which restores cellular communication to the level that was at the embryonic stage, enabling cells to rebuild damaged tissues and organs, restoring youth and functionality. Speaking to us on behalf of BioChange, very pleased to introduce Ishay Attar, who is the founder, chairman, and chief technology officer of BioChange. He has led the company from an idea to a well-established, fast-growing company. Ishay has 20 years of experience leading the development of innovative technologies that address large unmet medical needs, such as tissue reconstruction, surgical repair, diabetes, glaucoma, dry eye syndrome, and COPD. Ishay has a successful track record, founded 4 biomedical companies, achieved 2 exits, LifeBond and Outsense. Those companies were acquired by large tier 1 corporations. So great pleasure to introduce Ishay Attar from BioChange. Ishay, thank you for joining us, and take yourself off mute and begin the program.
Today, I'm Ishay. I'm the founder, CTO, and chairman of BioChange. We are restoring body functions and tissues which are lost with disease or age. And we're using an innovative foam scaffold technology to do that. And I will talk about our technology named cell foam today. Actually, what we're doing, we're growing a young tissue in an adult individual. And we're doing that by looking at the embryonic phase of life and trying to mimic or resemble the communication between cells which are applied in the embryonic phase of life. So just in brief, BioChange developed the cell foam technology, and we turned it into 2 clinical products which are on the way to approvals and clinical studies. First one is the ReGum for treating periodontal disease and other dental defects, and the Revolette, which is intended for skin regeneration. In parallel to that, we have brought to the market 3 veterinary products, which are based on the same technology. The ReGum Vet, again, for treating the same disease, which is very prevalent in humans as well as in pets. And also a vet foam for treating urinary incontinence, and another product for achieving hemostasis in severe injuries. So we're dealing with extracellular matrix here. Extracellular matrix is super important. Actually, it's a 3-dimensional network of proteins, mainly collagen. Mostly tissues are around 20% cells and around 80% matrix. So most of those tissues that we see and feel are made of the matrix, the ECM. It's not only providing the structural support, but it also facilitates communication between cells. And there's a lot of research, very recent research, and new information being uncovered about the role of the ECM, especially at the embryonic phase, and in wound healing and in recovering from regenerating tissues and organs. So the need we are addressing is finding a replacement biological scaffold that functions more or less like the young extracellular matrix. So I mean, most of the groups or researchers in our field, they're trying to mimic the natural scaffold of tissue, but they're trying to mimic the adult natural scaffold. And as you know, adult tissue, especially humans, but in many other mammals, they don't regenerate very well. We lose that regenerative capability, or if we can even call it the generative regeneration of tissues and organs, we lose it after the embryonic phase, after actually the beginning of the embryonic phase, which is called the organogenesis phase. So we're trying to bring it back to the patient, or to not even a patient, an aging person, just to regenerate and recover those tissue, and kind of we trick the cells to believe in the adult, we trick the cells to believe that they are temporarily inside an embryonic tissue. That's what we are trying to achieve. We have a platform technology, and we're going with it, as I mentioned, first of all, to the pet care market. We're already there, very active in revenue stage, commercial phase. Next, we'll be in the dental arena, and after that, in aesthetic and plastic surgery. And after that, we'll go to urinary incontinence, neural repair, macular degeneration, skin graft and wound care, orthobiologic, and finally, we want to get to limb regeneration and full organ regeneration. In all of the bullets that I've mentioned, we have proof of concept, but not yet in limb regeneration. This is something more futuristic that we aim to be there in the farther future, hopefully. Our strategy, as I mentioned, is to bring the veterinary product to the market, help the pet or the animals, also validate the product, de-risk them in the veterinary market. Everyone benefits from this strategy. We will have around 500,000 revenues this year, 2 million next year, and growing. Furthermore, we are able to bring our periodontal, the REGAM product, a periodontal disease product, to the market, to the FDA approval with a 510K. It's a quick approval because this is a medical device. Just the scaffold is a medical device. We don't provide it with any cells. And as such, it can be qualified for 510K quick process with FDA. Not same for the aesthetic product, the Levolette, which is an injectable. It's injected under the skin, and that will be a PMA product. This is a video that just demonstrates the cell form in one of its forms. In this case, it comes as powders. They are mixed at the bench top and can be injected into any site inside the body. And it takes a shape, it stabilizes, and within a few minutes, it becomes insoluble and just stays there for a few months. The body then works with it. It has very unique biomechanical properties. It has an abundance of integrin attachment sites. It means that the cells, any kind of cells, especially fibroblasts, are attracted to it, and they know how to attach to it with those integrins that they have. The form is also unique in the structure. It's a closed cell form and not like a sponge. It's not a sponge, actually. It's very different. The biomechanical properties are also very similar to the tissue. And you can see it very nicely in this video here that's being presented. So why do we use gelatin as the main structural protein? Why do we use gelatin? Because it's very close to collagen, but it's actually less immunogenic than senogenic collagen. It's much better. It's regulating the immune response to M2, to pro-regenerative. So gelatin has also been used for 75 years as a biomaterial, as an implant in various hemostats, or other implants. So the FDA and the other regulatory agencies knows it very well. It's also reversible. There's an FDA-approved collagenase we can inject and then reverse the implant, just dissolve it immediately if we want. Why the enzymatic cross-linking that we use? Because it's very biological. So we developed all those components to make the perfect foam scaffold that is I'll talk about the form in the next few slides because that's the most interesting part. But just to finish up about the enzyme, so the enzyme knows how to stabilize the gelatin very precisely, no toxicity, and it's very easy to inactivate it and remove it after it's done cross-linking. So why foam? I guess this is the most interesting part of the lecture. So what you see in this video is a real live imaging of an embryo of a zebrafish developing from a ball of cells into a fully formed organism with all the multi-level complex plan. Everything just forms within 2 days, just 2 days. So we'll see it again. So how is this happening? How is this smart material that's called tissue knows where to grow, how to grow, what to become, and to form a shape. Where is the information about the shape? So obviously it's in the DNA of the cells, but how do the cells know to communicate that information between each other? In the human, by the way, it takes about 6 weeks to build all those structures. Okay, it's a little bit slower, but we are all doing it. In the beginning of life, we all knew how to develop new tissue, new organs, and we lose that ability once we become grown, full-grown organism. Actually, even a just newly born baby is already doesn't have the regenerative ability of the embryo. So how is this happening? So each cell in this embryo is actually, it's an independent, intelligent entity. It knows to receive signals, it knows what to do with those signals, and it knows what to become and where to grow. Recent research uncovered more of that way that's communication between the cells. Especially I'm pointing out the research done by a group led by Professor Geo Campas from UC Dresden. So he injected very small micron level ferromagnetic oil droplets into the extracellular matrix of that zebrafish when it's growing. And he measured the mechanical forces that the tissue returns back on those ferromagnetic oil droplets. And what you see here in the right is actually a live imaging of the mechanical forces going on in the embryo, and he discovered that this is actually a foam. I'm going to show you another really interesting video.
The way in which an amputated.
So there's several animals that can regrow an arm when it's amputated. The best one is the favorite, and the winner is actually the axolotl, a type of salamander. So in salamander, if they lose an arm, the arm will grow back. It will grow back so perfectly that you can't even see a scar. You can't even know where it was amputated. How and what's going on in the arm of the axolotl? So actually, this at the beginning phase, the arm is resetting the biology. The beginning structure there is called blastema. It's very similar to the blastocyst of the embryo. And then it just grows. It just knows the original blueprint of the arm, where to go, when to make this elbow, when to make the fingers, everything just grows perfect. And it can do it 5 or 6 times within the lifetime. So we look also at the axolotl trying to understand and learn what's going on here. Another animal that we should maybe point out and learn from is the spiny mouse. So there's a lot of research going on spiny mouse, because that's a mammal that can grow the arm, not the full arm, but just the end. And we found out, really not us, but researchers found out that the extracellular matrix is a critical regulator in the wound repair in this specific animal, in this specific case. And I believe that it's not a matter of if, but a matter of when we can actually improve in regenerating limbs and organs in humans. And the cell form that I will show you more of our data in a minute is a groundbreaking technology designed to let nature do its magic. So just as in the embryo, just as in the amputated arm of an axolotl, we are working with nature to regenerate tissues and organs. First of all, we inject this to the skin. And as you can see here, this is an injection of cell form of the foam to the hypodermis of a rat, and it's a mesotrichome staining 1 month later. You see the ingrowth in blue, the ingrowth of tissue into the foam, and we see angiogenesis, new blood vessels being formed. Now, again, this is without any stem cells. It's just the scaffold. But we are the first ones to develop a scaffold which is in a foam structure. This is another example in a pig skin. The foam was injected into the left area over here, and degraded. The body just removes it within a few months and leaves behind engineered tissue. The engineered tissue that you can see here is new skin, so it looks very different from the control cell line. And we wanted to analyze what's the characterization of that skin. Okay, what is it made of? Is it a scar tissue? Is it a young tissue? And actually, we've done some immunohistochemical staining, and we've demonstrated that this is young tissue. It has type 1 collagen, it has elastin fibers, and it has the morphology of young tissue, not of scar tissue. Another tissue that we've treated is bone. We've applied cell foam into induced drilled cavities inside the mandible, in this case, or canine, and we've compared it to existing bone graft made from material called hydroxyapatite and also empty cavity. The results were also astonishing. We've expected that with a soft scaffold, we're not going to get so much bone regeneration, but actually we did see bone regeneration in a very, very exemplary way. You can see here the control of empty socket in B, and C, you can see the positive positive control product, the hydroxyapatite, and the cell foam. The cell foam generated very strong, dense bone around, osteons formed around the biomaterial. Not so with the hydroxyapatite. Since we're very active in the veterinary space, veterinarians have used it in proof of concept to show that if a bone doesn't heal, like in this case, a critical bone fracture doesn't heal, the mandible of this dog just broke, totally broke, and it didn't heal naturally with the fixation, so they inserted the foam in there, and within 8 weeks later, it healed very well. Our first commercial human product is going to be the Riga. We are addressing here a huge unmet need. About 40% of the people suffer from moderate to severe periodontal disease, and in this case, we actually see it's not just a tissue; it's an organ that's being destroyed over time because of that inflammatory disease. We see bone retraction and soft tissue retraction, and we wanted to cure both of them. We wanted to fix the tissue, to restore it to the original state. And that's what we are doing with our technology, with the cell phone. But just before I show you the results, it's also important to know that oral health, especially the periodontal health, is very, very relevant to well-being and longevity. There's a lot of documented information and data that actually shows us that periodontitis can significantly reduce telomere length. It can lower inflammation systemic effect. It can solve immune system dysregulation. And actually, there's a really big study that was done in canine, in pets. They took like around 350,000 dogs and compared those that do get yearly cleaning and those that don't, and there's a 20% decrease in risk of death with those that receive annual periodontal cleaning. So the first thing you can do when you hear this is just, go get your gums cleaned every year. But when we are going to get to the market, we'll be able to actually restore that and solve and cure that incurable unmet need. In a certain study that we've done in canine, we've inserted three-dimensional shapes that fit the canine story into the gums, and it's done in an open flap surgery. The flap is being removed. It's a small surgery. The foam is inserted and then sutured over it, and the results were recorded and published in Frontiers. We didn't do the study. The study was ran by key opinion leaders, veterinarians from Europe, and we saw bone regeneration. We saw elevation of the bone line, which is something which they haven't seen before. We've also seen recovery, 100% improvement in the soft tissue recovery as well. So we recover the bone and recover the soft tissue. In a longer follow-up and some other case studies, we see more and more that data. We currently have hundreds of cases already treated and followed up, even up to 2 years. Our next product to the market is the Revolette. Revolette is a class 3 medical device. It's going to be used to rejuvenate the skin. So we lose about 1% of the skin collagen every year. And it's irreversible regardless of various products that claim to help. In this case, we've designed the foam to be injectable into the hypodermis, and this can be used to rejuvenate and recover the collagen of the skin. It's also, actually, I'm not going to take long, but it's a huge market with very strong growth. We've compared it to existing products. There are 3 existing products in the market, other scaffold that claim to do regeneration of collagen. And we've compared in head-to-head study. And as you can see, that you already know that the amount of blue resembles the recovery of the collagen in the skin. You can see the other competing product, and you see what we can do with our cell foam technology. It's going to be give dermatologists a really effective tool to recover the skin. Another interesting application of our technology, and also relates to the previous lecture, we can also enhance the engraftment of stem cells into the tissue. We've done some studies where we inject labeled cells into the skin, either without the foam or with the foam. They don't survive. As you know, stem cell or any kind of therapeutic cells don't survive very well when they are injected into the host tissue. But here, when they're injected with the foam, they survive very well. And we are interested in collaboration with cellular therapies companies or startup companies or researchers. They can use the cell foam together with their cell to enhance engraftment. We have done another experiment together with cells on macular degeneration. Macular degeneration is the leading cause of blindness. And in this case, the macular regeneration is the RPE layer that is being lost. RPE cells are the cells that are in charge of maintenance of the photoreceptors. And once they are degenerated, then the photoreceptors, they die later, and the blindness begins. So in this case, when injected the cells alone, those are engineered cells made of fat. And they didn't help very well, but once injected into the subretina together with the cell foam, we cured this disease, in this case, in this model. So this is also very promising application of our technology. We have already 18 granted patents, and I want to thank my team, and I'll be happy to answer questions.
All right, Ishay, thank you very much. Very interesting presentation. So just to be clear, the gelatin does not always have to be combined with stem cells. It may be combined, but it can be delivered without the stem cells. Is that correct?
That is exactly correct. We didn't do a lot of work combining with stem cells. We want to do more work, but currently our strategy is to develop products which are based solely on the foam, which is a medical device. It's not a biologic, easier to get around through the regulatory authorities.
Okay, so since it's classified as a medical device, the gelatin, I would imagine, does not have to be customized to each patient. It's just the same gelatins delivered to any dog or to any gums.
Yeah, exactly. It's not customized to patients. It's not even customized to tissues. It's just restores the communication between the cells. So the cells in the embryo, they know how to talk through that foam. They know how to talk with each other, and they know where to go and what to become. They even do it in the arm of an amputated salamander. Okay, now a salamander, when it amputates the arm, there's a blastocyst, a blastema, sorry, which is formed there. So they know how to restore the original blueprint of that organ. And how do they know it? They know how to transfer information between each other. And that's what we are doing. We're allowing them to do their job, and they do it the natural way. So we don't need to customize it.
And then for the dental industry, the gum product, have you done any work to try to get reimbursement lined up with insurance companies, or is that too premature? So just from a timeline point of view, you're generating about $500,000 this year from pet.
Yeah,
pet
we just
products.
started. We've just started with sales. Yeah,
And then how long will it take to get the gum product on the market?
2 years, 2026, probably the end of it. Yeah, but 2026.
And then the skincare product, Revelet.
Yeah, so that will take longer in the US. It's a PMA, so it will 2027, 28 in the US. We can probably be there before in some other territories like Israel, Europe, Brazil.
And then for the gum product, is it envisioned that any dentist can use it, or does it have to be a periodontist or another specialist?
No, the periodontist will use it for restoring the gums, but it can also be used to restore bone. There's no need to use a GTR membrane. You know, usually when they want to restore bone, like to strengthen the bone for implant surgeries, so they have to use bone graft and they have to use a membrane. The membrane, they call it guided tissue regeneration membrane because it prevents the soft tissue from interfering the bone tissue because it grows 10 times faster than the bone tissue. So they have to put a barrier in there. In our case, there is no need for a barrier. You just put the foam there and the cells, they know what to do.
So is it faster for the periodontist or dentist to install the gum product, the gelatin product? Is it faster to do with your product or the existing process?
Is it faster? I didn't understand the question. So.
Yeah, I'm just thinking about the economics from the dentist point of view. Is it easier to work with?
Yes, of course. You don't need to use two products. You only need to use one product instead of two. It's less expensive, and there's several indications which are currently are unmet. You cannot recover the gums. If you lose the bone, you cannot recover it. It's only very specific cases that bone recovery can be achieved with current bone grafts and tissue grafts. And usually they stop the deterioration for a while or for sometimes even for a long time. But they cannot rebuild the lost tissue. That's currently impossible.
And then all the other products that you might bring to the market after Revelet, after the skin product, do you plan to build this company using direct salespeople or distribution channels, or to license out all those patents you have to the different fields of use?
Well, we will do both. In the veterinary market, we distribute to global distributors. For instance, in Europe, we're partnered with one of the largest distributors, IM3, largest distributor in the field of veterinary dentistry. We will do the same in the humans when we get there. We'll probably try to partner for distribution. And in the case of, if we do it, collaboration, we are looking for collaborations with cellular therapeutics companies. In this case, we can provide it as an OEM product or to do it together or some other way, or license.
Okay, currently is BioChange doing the manufacturing, or do you outsource that?
Oh yeah, we are currently manufacturing it in our company in Yokneam.
Okay. Is there any
We have
is there any supply chain issue you have, or any concern you have about any of the compounds you put into the gelatin, scaling issues?
Yeah, we will need to invest into GMP manufacturing of the enzyme that we use. It's a recombinant technology that produces the enzyme in E. coli. We will have to invest in that when we go into when we go into the the human market later on.
And I would imagine some of the patents are composition of matter patents,
Oh yeah.
and then some are maybe process patents as well.
Yeah, we have methods and mostly composition of matter patents.
And it's a pretty good geographic coverage with the patents and covering U.S., Europe, China.
Yeah, we have China, Japan, South Korea. I mean, granted patents. U.S. of course, Israel, and Europe, and we have also filing in Brazil and other countries. Less common, but we did get claims granted in most of the territories, and it's still ongoing. You know, we have many still pending patents as well.
Yep.
Too. Yeah.
Is there anything you want to say about the capital intensity or capital needs you have, or capital raising plans?
Yes, so we are now open in round A funding. We are raising between $5 to $10 million. It depends, and we want to use this to either bring one product or two products to the market. $5 million is the cost of bringing a product to the human market. Some of it will be funded from the revenues from the veterinary products. I have to maybe say that our margins are very high. We have good margins on our veterinary product. We'll have even better margins on our human product. So we are profitable from the first product. We don't need to scale to be profitable. And it's a very big advantage and ease on the cash flow. But we do need to raise $5 million to get the ReGum product to the human market through the FDA and to commercialization. Yeah. And we will also raise $10 million if we can.
Well, it sounds profitable. It sounds like producing the gelatin is very doable and very high value add to the veterinarians and dentists that use it.
Yeah.
So that sounds good. Do you think research and development expenditures will start to decline a little bit now? Have you done most of the research you need to do?
I mean, not really. We are an R&D oriented company. We continuously do research. You know, we even do research on recovering a full limb or an organ. I haven't shown data about it because it's still in the research, but we're doing we're investing a lot of efforts to understand what's going on. Okay, how can we rebuild or actually stimulate the body to rebuild itself, tissues, organs, you name it. And I hope we can get there. This is our dream and passion. Okay. So we are oriented on R&D. We're not going to reduce R&D expenses. Hopefully we'll just increase them over time because it's a lot of work.
Okay. Another question came in. Do you want to tell us about your board of directors or advisory board, and how any of these particular individuals may help you get into these markets or get aligned with a large distributor or something, anything like that?
Yeah, so we work with a line of consultants that we have a line of very top tier consultants in medical, aesthetic dermatology, dental, obviously, and also business and IP consultants. We work with the top of the top, and this is all I can answer for now.
Okay. Okay. Well, great. I don't see any more questions. So thank you very much. Is your contact information here, or I guess the website's just BioChange?
Sorry, I need to let me quickly put it in here. I forgot. You're welcome to contact me. There we go.
Yeah, so the website is biochange.life.
Right, biochange.life, and my email is Ishay, ISHAY, @biochange.life.
Okay, well, great. Yeah, I think we exhausted all the questions. So Ishay, Attar, thank you very much for introducing us to BioChange. Very interesting company, and wish you a lot of continued success.
Thank you very much, and thank you everyone for listening. Goodbye.
Bye.